protein itself is present in the cell and is acting as a transcriptional coactivator. Only this name means that we actually do not really know what it's doing. It's doing something, it's helping transcription. Yeah? The protein comes in two flavors. It comes in a long version, which, now I see it. Okay, so the long version of 75 kilodalton has this integrase binding domain at the C-terminus, and this is the part where the HIV integrase binds. Yeah? The short version, 52 kilodalton, doesn't have this domain, so this doesn't act as a cofactor. Only the long version acts as the cofactor of HIV. And then, even more importantly, at the N-terminus there is this PWP domain, and this could be an exam question for students in biochemistry. If you know what this means or what it could refer to, it's the abbreviation of the amino acids, the proline, the tryptophan, and the proline. Because these are present in this domain and they form an aromatic cage that recognizes specific features on histones. And you probably have heard about epigenetics. Genetics, that's our code, our sequences. The epigenetics is the modification of this code by methylation, acetylation, that makes the chromatin open or closed. Yeah? LADGF is recognizing a specific mark, I will talk about it later, that is leukemia MLL protein. There is all these names of these proteins, and the leukemia MLL protein, there is all these names of these proteins, and the interesting thing is that these proteins are also involved in human disease. So after we discovered this protein in the context of HIV in 2002, 2003, we later discovered that the same protein is involved in other diseases, and that's why in the introduction it was mentioned that I also study leukemia, I also study Rett syndrome. These are diseases where the same protein also plays a role. And we do there also drug discovery for these diseases. So people with an interest in neurodegeneration or in neurodevelopment and leukemia, we also do that in our lab. But today I only will talk about HIV. Okay. So this is the function of the protein, and it's actually an experiment we did years ago to prove how the protein works. This is the viral DNA, this is the bolus, the integrase, and so the LAGF has these two domains. It has this head of the N-terminal, which recognizes the DNA, and it has a C-terminal that binds the integrase. And so on the right you see the experiment that we did. We actually overexpressed C-terminal domains, so the tail of the protein, and the tail can bind to integrase, but since it cannot recognize the DNA, it will interfere with the normal process. And so when we made these cell lines overexpressing the C-terminal truncations, we got a block of HIV replication, which was proving at that time that LAGF has this important function for HIV, but also that it could be seen as a drug target. If we could block the interaction between LAGF and integrase, we could block HIV replication. Yeah? And in a sense, LAGF, you can see it as kind of like the rope that is bringing the ship, which is the integration complex, is the virus, into the harbor, which is the chromatin. So it tethers, it brings it just to the chromatin where it can integrate. Okay. So after showing that this protein was important for integration, after showing that it was a potential drug target, Peter Cherepanov, a former postdoc in my lab, he solved the crystal structure of the complex. And you see that right. So the integrase has these three domains. It has a catalytic core domain, which is the catalytic function, and that domain is interacting with the C-terminal domain, the IBD of LAGF. And if you see here the crystal structure, the integrase catalytic core makes a dimer, and here there is this interface, there is a pocket. Well, the IBD is a 4 alpha helical bundle with an interhelical loop, and this loop is protruding in this pocket. Most drugs are targeting enzymes or receptors, the drugs that are used in the pharmacy. Yeah? Protein-protein interaction are difficult to make a drug because often it's a flat surface and you need a lot of binding energy to overcome the interaction. Yeah? But in this case, we see a very defined interaction with a loop and a pocket. So we gave this crystal structure to a molecular modeler, a PhD student in my lab, and he did what we call structure-based drug design. So he actually drew molecules, we call them pharmacophores, to fit into the pocket. And we know the amino acids, so we know there is an aromatic amino acid, a charged amino acid, so he can make like a molecule with rings, aromatic rings, with a hydrogen bridge, a kind of theoretical, a virtual molecule. And then he looked into the libraries of commercial vendors, often from Eastern Europe. People, chemists, made a lot of compounds, and they are available, you can buy them. And with the computer program, he looked for lookalikes. He looked at molecules that resembled his virtual ideal molecule. And he found about 20 that more or less could fit this ideal pharmacophore. We ordered them, we dissolved the compounds, and we tested them. We had an assay in which a vitro assay, which we looked for the interaction between integrase and LegF. And yes, indeed, some of the molecules could block the interaction. We gave the molecules were not active yet at that stage to block HIV in cell culture, so our chemists optimized the compound, so they made modifications. We tested again in the in vitro assay, they were more potent, more powerful, and eventually we got compounds that also block HIV replication in cell culture. Yeah? In the end, we also got a crystallographer in our team who could actually soak the compound in the pocket, high resolution, and then you can really fine-tune hydrogen bridges, length, charge to even optimize the compound further. So that's what we did, and by doing that, we developed, and this is a publication from our group in 2010, what we called Legends. So these are small molecules. The structure is shown on the right in the yellow here, and actually this is the crystal structure. So you see that the compound is sitting actually in the pocket, and all of these Legends, they have a carboxylic acid, which is actually competing with an aspartic acid in the loop. So the loop is protruding, has a carboxylic acid, and the compound is actually mimicking that and is competing with this loop. So when the compound is bound, the IBD, LegF, cannot bind, and HIV cannot be tethered to the chromatin, and so you block HIV replication. Yeah? We call this compound Legends because they block the interaction between LegF and integrase, and also because it's a bit legend in the name, and yeah, you always have to be ambitious, so we hope they can be legendary compounds. Okay, so we published this, these compounds were other groups and companies made analogs of these, and there have been clinical trials, and I'm really willing in the question session maybe to answer some of these questions on that. Okay, but now I'm going to the real topic of today is, what about HIV cure? I guess you may know the issue and of the problem of HIV latency. So in the untreated patients, they have a high viral load, is shown here in the y-axis, and you see that a lot of these cells, they are red, they are productively infected. It means that HIV is producing new virus particles. Many of these lymphocytes will die every day, and every day there will be new viruses made. Luckily, over the last 20 years, we developed potent antiretrovirals that block HIV replication, and by giving them in a combination, a cocktail, we can actually block HIV replication, so that we come to the next stage where there is almost no virus detectable in the blood of the patients, but since the virus integrates in the genome of these lymphocytes, and some of these cells become memory cells, and this immunological memory you keep for the rest of your life, that's the meaning of your memory. So it means that some of these proviruses will stay latent, so it's no proteins being produced, it's not recognized by the immune system, but they stay, these proviruses, for the rest of the life of the patient. Yeah? And if you, that's then the final stage, if you stop the treatment, the memory cell that recognizes his pathogen, where he has a receptor for, will be activated, and then also the provirus will be activated. So if you stop the treatment with a patient in a couple of weeks, months, the virus rebounds and he gets productively infected cells again. So we cannot stop the treatment. That's why these people have to take these pills for the rest of their life. Yeah? And that's a burden, because you every day remember that you have a disease. HIV/AIDS, there's also like a stigma, there's a taboo. You don't tell it when you go to the bar, hey, I have my pills, but do you know I have HIV? It's kind of difficult. If you stop the treatment, you can be infectious again, also for your partner. You can have resistant virus. So if you talk with the patients, they want actually to be cured so that they don't have to take the pills, that they're not infectious anymore. And we retrovirologists, I mean, I went to the age 86 where we had to discover the virus. Then we went to the age where we had to develop antiretroviral treatment, and I was involved in NNRTIs, in integrase blockers, in combination treatment. Now we are at the era where we have to find a cure, to try to really find a solution. For the hepatitis C virus, which we developed antivirals later, we have a cure. We can cure hepatitis C. We cannot cure HIV yet. Okay, so that's the context, why we study this. Okay, so different strategies are being pursued to try to cure HIV infection. I told you about the Berlin patient, that's stem cell transplantation, only available for a limited number of patients, and high mortality. A person which is treated well and doing well, you don't take the risk of 20% to die. You don't do that. So that's impossible. Some people do gene therapy. You know, with CRISPR-Cas, we can cut out HIV. There the problem is we don't even know where the virus is hiding in the body. How can we deliver the CRISPR-Cas to all these places? There's definitely progress. It should be pursued, but it's still a lot of challenges to overcome. And the two main approaches that are being used are shown here, which is the purge and kill and the block and lock. In the purge and kill approach, the idea is the latent provirus is latent. It cannot be recognized by the immune system because it's not active. Let's give agents, compounds, drugs that activate the virus. So we purge the virus out of its sleep so that either the cell dies because of the toxicity of the virus, shown here, or the immune system recognizes these activated cells and then kills these cells. So you will purge or shock and then kill. So that's the purge and kill or the shock and kill approach. And many researchers doing that, or clinical trials on this, so far, but there is some advances in this, but there is no miracle yet. But it's an approach that people are taking. We took another approach, which we call the block and lock. And to be honest, so we published the first paper on block and lock, but the block and lock strategy was first coined by Susana Valente. She developed a TAT inhibitor. She gave it to chronically infected cells, and she reduces the HIV expression. But then she saw when she removed the compound, even in mice, that there was a delay in the rebound of the virus. So somehow, this TAT inhibitor gave an epigenetic imprint, maybe by DNA methylation, so that you could remove the compound and you still had this lock effect. She called it the block and lock. I will show you during the rest of the presentation that the legends, remember what I told in the introduction, can be used for a block and lock strategy. And so the idea there is the opposite of the purge and kill. If we find a way to put the virus permanently in a deep sleep, so that it does not get activated, even if we take away the compound, take away a drug, then we should not bother. Then maybe there is some virus somewhere, but it's not active, it's no problem. They say, oh, it's dangerous because we need to eradicate the virus. I don't think so. I mean, you maybe heard about retrotransposons and line 1 elements. I mean, our genome is full of old, ancient retro elements that were previous retroviruses, but that actually we overcome. And they became silenced, they became part of our genome. So having a piece of genome, I mean, a lot of the retrovirus also defective. So you're not worried about that. So I think it's conceptually a possible strategy. Okay. Okay. So where does the hypothesis come from? So remember that I told you that this Lejeff has this IBD and this PWP domain, and that the PWP domain recognizes the specific mark. So now the mark is mentioned. It's the 36th lysine, the K stands for the lysine, present on the third histone. And you know that the DNA is wrapped around nucleosomes. Nucleosomes are composed of histones. Histones have lysines. Well, this 36th lysine is triple methylated. This affects the charge, because the lysine is normally positively charged, and this affects the chromatin compaction. Yeah? Well, this particular mark is recognized by LegF. LegF binds, and this mark is present in active chromatin, where transcription takes place. So HIV, it's my enemy. I want to cure people from HIV. On the other hand, I kind of admire the virus, that it evolved in a way that was very clever to overcome all these difficulties in the human cell. I mean, this virus evolved to take this LegF protein, which is a molecular tether, to use it, to abuse it, to hijack it, to get guided to the chromatin at sites where there is active transcription. By doing that, ensures that when it integrates, it has a high chance that the virus will be transcribed, that the virus will make new babies. So the virus wants to get replicated. And it's ensuring that by taking the GPS, we call it the GPS, LegF is actually GPS, it's directing HIV to the best spots of the genome. Yeah, and I think it's a fantastic evolution. But knowing that, you can also say, and we did these experiments, actually Angela Cuffey did these experiments a long time ago. If we knock down, we deplete LegF from the cells, we see that the virus shifts to other sites. It's not anymore in these active genes, it's more in silent genes, in heterochromatin. So now we say, if we now try to repeat this with our legends, what will happen? Okay, I come back to the legends as a tool to study the hypothesis. Explain you that we discovered these compounds as inhibitors of the integrase-LegF interaction. It's a bit more complicated because these compounds, just by sitting in the pocket, they already interfere with the integration. We call this the allosteric effect. Thirdly, and this was later found, by sitting in the pocket, they also interfere with the production of new virus particles, with the assembly. So we have an antiviral which has an early and a late effect, which is a kind of combination in one compound. And then 4, and this is the novelty, the thing we published, we showed, is that the legend can also retarget integration, so bring the virus to other sites where it's inactive for transcription. Yeah? And that's maybe the most interesting mechanism because that can lead to a functional cure. Okay, so what we did, we answered 4 questions experimentally. The first question is, if we take cells, we add the virus, but in the presence of legend, will the virus integrate at the same location or a different location? Secondly, can we see where the virus is in the nucleus? 3, if this virus is at a different location, is it still transcriptionally active? And if we wash away the compound, like Susan Valente did, what happens? Does the virus pop up again? Can we reactivate the virus, or is it still silenced? And these 4 experiments, Leonard, who is a PhD student in my lab, he actually performed. Okay, so the first experiment, look to the right, because that's a bit the summary. We have this technique where we measure the integration site. We sequence part of the HIV genome and part of the cellular genome, and we know the junction, we know where the virus is in the genome. And then we do the same in the presence of a legend, and we see again where is the virus. And so typically the virus is for 60–70% in active genes, and if we add legends, we see that this is reduced up to 50%. So we shift away the virus from the active genes to the inactive genes. So we do retargeting of integration. So the first question was answered positively. And the second question, for that, I don't know if you can see it carefully, but we use a reporter virus, and the virus integrates, and we have an endonuclease that makes a cut in the provirus, and the DNA repair proteins will be activated where you have antibodies for these, and so we show where the virus is in the nucleus. And so on the right you see these cells in green. This is the nucleus each time of an infected cell, and this I don't know if you see it, but the orange dots are the proviruses. And so we measure the location. So we measure the virus has to come into the nucleopore, in the nucleus, before it integrates, and we measure the penetration depth. How deep does it get into the nucleus before it integrates? And we make this distribution plots. So the 0 is a virus which we integrate at the nucleopore, and it goes until like 8, 10 micron in the cell versus the center of the nucleus. And so what do we see? If we knock down LegF, we published that before, we see that the virus drifts more to the center. So the virus comes in, it wants to see LegF as a GPS, there is no LegF, so it starts to drift and it takes longer, and it moves, it diffuses longer to get integrated. And we do the same experiment with the legend, and we see even a stronger shift towards the nucleus. And we could control the experiment to the right, because we have a virus which we made resistant to the legend. That is 2 mutations, so that virus is not shifted. So it's a really specific effect of the legend. So we know that if we add legends during infection, the virus is retargeted. It's also at a different location in the nucleus. But the big question is functional. How does this virus behave? For that, we did use this double reporter virus made by Eric Verdin. It's an HIV virus which has 2 reporter proteins. The red protein, MK02, is driven by a housekeeping promoter. So it means that if you infect the cells, all cells will produce the red protein. So all infected cells will be red. The green fluorescent protein, shown there on the bottom, is actually driven by the LTR of HIV. It means that if the LTR is active in a productive cell, the green protein will be produced. If the virus is silent, latent, there will be no green protein. And so we can fax these cells. So we take cells, we infect them with reporter virus, and we measure the green and the red fluorescent protein. We put them in the quadrants, and then you can see that you have these proteins that are red fluorescent protein positive. They are infected. And the ones that are on top also green. So red plus green makes yellow. So the upper right quadrant, these are the productively infected cells. And so by comparing the red cells, the quiescent cells with the productively infected cells, we know the ratio between the activity of the promoter. How much latency do we have? And so we infected the cells with this double reporter virus in the presence of the legends. Now we have to becomes a bit complicated. Legends are antiviral, so they also inhibit integration. So you will have less provirus after addition of legend. That's sure. But we're interested in the residual provirus, the provirus that still integrates in the presence of legends. Is it active or is it latent? So that's why we look for the red and the green, and that's shown here. So on the top, on the right, you show the DMSO control. And you see, it's hard to see the numbers, but I think it's about 20% of the cells that got infected. So it's the red ones, and then the productively 5%. 20% are infected, and actually 15 out of 20, so 3/4 are quiescently infected. And that is also shown on the left. You see about 70% of the cells are quiescent. If you do the same experiment with a high amount of legend, 50 micromolar, you see much less infected cells because inhibition of integration. So in total about 5%, I think. You have to add those 2 numbers. But very few of those residual infected cells are productively infected, all quiescent. It's 5% and 0, 0. So if you put this ratio of quiescent, you see it in the y-axis, you go up to 100%. So the cells that are still infected with the provirus, all the provirus is retargeted, and all the provirus is actually transcriptionally inactive. So we made a lock. We locked the virus in a latency. Of course, the question remains, this is in the presence of the drug. If you now infect your cells, and you wash away the virus, and you wash away the drug, and you wait 2 weeks, then the virus will be silenced, part of the virus. If we now reactivate the virus, we push it again to activation, what happens with the retargeted provirus? Can we still reactivate it, or does it stay latent? And that would be a lock phenotype. Well, we saw the results. Okay, and so what we measure is actually how many of the quiescent cells on the bottom are moved into the active cells. And that's shown in the plot on the right. So we show the fold increase in the GFP positive population. And at the 0 value, legend 0, shown here, you see that we get about a 2-fold reactivation. So we get a 2-fold increase in the GFP positive cells. But if we had treated the cells with the legend during the infection, and washed away the legend, we see that it drops until about 1-fold, meaning no activation. And the same is true if you look at the reduction in the quiescent cells, 30% reduction, so it means that these cells are activated, well, less than 10%, 5%, if we had pre-treated with legends. So it means that if we add legends during the infection, we retarget the provirus. It's at another location in the nucleus. It is transcriptionally silent and is very difficult to reactivate from latency. And that's what we call this block and lock phenotype. Yeah? And so we propose that these legends can be used for a functional cure, because if you would add them during the infection, you would get a virus which is blocked for transcription and locked for reactivation. Okay, I don't know how much time I still have, because we started late. 10 minutes? I will summarize. Okay, so, okay. So this is the story of the block and lock. I just now briefly, and these papers are published so you can read them, but they're not so easy to read, and that's why I spent time in the introduction. 2 PhD students, it's PhD level research in my lab, Gerlinde van Sant and Julie Janssen. They used 2 techniques to understand the mechanism of what happens. So you've seen that we retarget integration from 70% to 50%, but this is not to the random control. And still, we have a very potent phenotype. So the phenotype is stronger than the genotype, so something else must be going on. And they used 2 different techniques. In the first paper, we used barcoded virus, which means that we make an HIV virus with an individual barcode, a specific sequence, and the sequence is in the provirus, and the sequence is also in the mRNA transcript. So we infect cells with the barcoded virus, we measure the integration site, so we know where each provirus is, and then we measure how much mRNA the virus makes, so we can make a link between integration site and transcriptional level. Yeah, so at that moment in time, nobody knows whether an integration site, the place where you integrate in the genome, makes a difference. Yeah? The only way to do that is to try to find the correlation between integration site and mRNA. And that's what we did with this barcoded virus. The other technique is a fancy, very sensitive in situ hybridization, where we measure the viral DNA and the RNA. And again, we try to correlate how much RNA with the provirus being present. Okay, this I will skip. This is the most important slide on this. Is we show the RNA expression for each barcode. Each dot is a barcode. So it's a provirus at a specific site in the genome. And so we see that increasing concentrations of legends reduce the overall mRNA transcription level. This is for viruses that integrate it, because otherwise we would not have a barcode. So it's not due to the lack of integration, that's controlled for. It's for the viruses that we still have less transcriptional activity. Yeah, that's clearly shown here. And even at the right, you see that if you look at the specific barcodes that have no RNA, you see that the number of them increases upon increasing concentration legends. So lots of these proviruses that are retargeted are in deep latency. Yeah? For each of the chromosomes, we have actually this pattern. So we have each dot represents a site of integration, and each vertical line means how much mRNA at this site. Yeah? So how transcriptionally active is the virus? And then you see that increasing concentration of legend reduce the number of peaks because you have less integrations, but per peak also the transcriptional level goes down. Although you see few sites where we still have high transcriptional levels. So it's not a complete mechanism. There is still some leakage, and I will explain at the end why we think this is happening. Okay, so that's another important finding. I told you that LegF recognizes the specific H3K26-methyl-3. We can actually determine the distance of the provirus to this mark. And so we see that in the absence of legend, the barcode that is transcriptionally active or not active, there is no difference. It's non-significant here, meaning that all viruses that integrate are close to the LegF mark. Yeah? And this is even a physical distance. Yeah? But if we increase the concentration of legend, we see that there is a huge difference between the distance from the viruses that are active from the viruses that are not active. So again, showing that we shift the virus away from the specific mark, and these viruses become latent. Yeah. If we look for the acetylation mark, which is a mark for an enhancer, we see that already without legend, there is a difference. The virus which is close to the enhancer here has transcriptional activity, while the virus which is further away from the enhancer here is not transcriptionally active. Yeah? So this is a second mechanism that takes place. And this leads us then to the model that I will show. Maybe think really what is happening. That is in the default, in the normal situation, the virus will interact with LegF and with the mark and get integration in transcriptional units. Yeah? If we add legends, we actually suppress this normal mechanism, and then the virus starts to drift, but still it can stochastically, by coincidence, integrate close to an enhancer, and then you will see still transcriptional activity. And that's what explains this residual peaks. So now we're working with enhancer blockers to add on to our block and lock phenotype. Okay, then the last part of the presentation is on this imaging technique. So here we use microscopy to look at individual cells, and we hybridize with the probe the DNA, and this is colored in red, and we hybridize the RNA with a probe that is colored in green. And so we count each time 100 cells, and then we count the number of dots in each cell to have an idea about how much provirus and how much RNA. Yeah? And then this leads us to this kind of, you can see it, it's quite a lot of light here actually. So I hope you can see it, that this is with the legend. This is in the unactivated condition. This is the activated condition. And so you see that this is a magnification, that in some of the cells, so definitely in the control, you see cells with green dots. So these are productively infected cells. If we activate the cells, you will see many more of those. So these cells are activated to produce a lot of RNA. But you see that if we increase legend concentration, less and less cells will have RNA, and the same is true for the reactivation. So we block and we lock reactivation. If we take a control, which is raltegravir, which is a known integrase inhibitor, but it has no effect on LegF, it's not doing that. It's also inhibiting integration, but you still see RNA both in the unactivated and the activated condition, meaning that we have this specific effect. And we could quantify this, shown here. So you see that each dot represents a cell, and we count how many RNA spots. For instance, this cell has about 30 RNA spots present. Yeah? If we activate with TNF-alpha, this goes up. More cells produce RNA. But if we add legend, you see that the average drops. Less and less RNA per virus. Yeah? We still can reactivate, but also this goes down. Yeah? So blocking and locking. Residual activity, again, probably virus close to an enhancer. Yeah? So our block and lock effect is about 90%, but there's still small margin that we want to improve. Okay, so we see the same effect in the primary cells. There we actually see some effect of selection, but I will skip it, lack of time. Okay, and then, yeah, the question is, we did these two papers with a research compound, the legend we developed ourselves in the lab, which is present at micromolar concentration. So people say, yeah, but that's a very high concentration. This is not clinical compound. So then we collaborated with Gilead and we used their compound, which is a nanomolar active compound, and we actually got the same results. There we saw the block and lock phenotype at a very low concentration in cell culture. Okay, so this brings us close to the end and some conclusion. When we tried to publish a first paper on the block and lock in 2016, we got a lot of skepticism. People saying even that it was unethical to propose a block and lock, because at that time people were trying to eradicate the virus and we were going to keep the virus, although in a latent state. And so it has gone back and forth until some papers from the group of Matthias Lichterfeld in Harvard. So Matthias is studying elite controllers. Elite controllers are people that can control, probably by a strong immune system, HIV replication, so that there is very low level of HIV in the absence of treatment. Yeah? And he looked at where the virus was located in the chromatin in these patients, and he found the virus in inactive chromatin, in heterochromatin. So probably the hypothesis that these people can eradicate the virus which is transcriptionally active, the virus which is located in the active genes, and the virus that happens to integrate in inactive genes is not active, is not eliminated, but it don't bother because it doesn't produce virus. So this block and lock is actually a bit of a natural mechanism which happens in elite controllers. And then more recently, Matthias did the same experiments in chronically infected patients under treatment, and there, over a period of 20 years, he also sees that there is an evolution of the provirus being present in inactive chromatin. So the idea is that our immune system is not inactive. It kills cells which are productively active, and it selects for virus which is not transcriptionally active because it's located in inactive chromatin. So our idea to facilitate this, to push the virus in deep latency, gets less and less critique because it seems to be something which is happening in the patient. If we could do that as a drug treatment, we would facilitate something which happens naturally. And so Matthias Lichterfeld, he contacted me, he cited us also, and he likes really the concept because what he's showing in the patient, I mean, we proposed actually in cell culture experiments. Okay, so that's where we are. And so in the summary and the timeline, so we discovered LegGef in 2003, Legends in 2010, proposed this block and lock in 2016, and now I'm really advocating clinical trials to see Legends as an antiviral and not Legends as a cure. It's something which is testable. We can discuss it in the question session, but it's possible to use Legend as an antiviral or as a PrEP during an acute infection where the reservoir is probably still formed. If you add it, we could shift integration sites, which would increase the deep latent majority of the virus. We could also do it as a first-line treatment or as a chronic infection and a treatment interruption. Okay, so these are the ideas we have also in the field to try out this concept of the block and lock with the Legends in clinical trials to see if we can achieve a functional cure. And with that, I would like to thank the people on the picture, the people who are now present in the lab, but I talked about 20 years of research, many people collaborated, there's a lot of collaborators, but I will stop here and thank you for your attention. I'm willing to take questions.
Hi, so the floor is open for questions.
Don't be shy.
Yes,
Yeah. Can you speak in the microphone?
Just the mic.
Yeah.
Thank you.
Okay.
Yes, Richard.
Since when you talked about the block and lock system, I assume that it's more like antagonism or something. So, regarding the legends, the question is that are there specific sites on the CD4 T cells, or should I say the virus targeted cells in the human body, where those legends bind to before they can function?
So legends are small molecules, so they enter each cell, so they don't have to bind. Yeah? They are not specific. They will enter all cells, not only the CD4 positive cells, but since legends do not bind LegF, they bind integrase, there will be no toxicity. They only will be active in cells that are infected with HIV. CD4 cells, dendritic cells, myeloid cells, all cells where integrase is, they will bind and they will block. So they are very specific. Not because they're specific for a particular cell type, but they are specific for the integrase. And we have no integrase in our cells, only when we are HIV. So that's not a problem.
So, since we are saying there will be no toxicity, my question is that are they not going to load up in the human body or cause anything bad in the future?
Mm-hmm. So for that, it's a good question. Before putting a drug from a cell culture to a patient, people do animal studies, mouse, dogs, primates, and then healthy volunteers. And what they do is PK, pharmacokinetics. So we add the compound to the patient, and then we determine in the blood how long the compound is present. And so you see clearance, because these compounds, they get in a cell, they diffuse out of a cell, they can be metabolized, and they can be excreted. In this case, probably because of the carboxylic acid, they are glucuronidated, and they excreted in the kidney, in the urine. So you actually, the problem with some of the legends was actually the opposite, that we had to short half-life. So that we had, because nowadays, I think you know, these anti-HIV drugs we give in a combination, it's 1 pill a day. But you need each active compound to be active during 24 hours. So you need a compound to work anymore. Yeah? And so with the legend was the opposite. It was clear too fast. Yeah? And so people had to modify the compound chemically so to get it stable during a day. Yeah? But it's a good question.
Any more questions? Prof. Mapura?
Okay.
Thank you for a very interesting talk. 2 questions. I should presume that this integration is site-specific. The integration is site-specific, or is it a random kind of integration on the human genome?
It's a good question. It is not sequence-specific, yeah, but it's also not completely random. So it has a preference for some bases in the genome, but it's a very low specificity. The specificity is not in the DNA sequence, the specificity is in the epigenetic recognition. It will integrate close to this H3K6-methyl-3 because there the tether binds. Yeah? So LegF, so the virus comes in the nucleus, the integrase binds LegF, LegF will target to this epigenetic mark, and then integration will be in the neighborhood, very close. Not sequence specific, because it will be something in the neighborhood, with some preference for some bases, some A's and T's and so on, but not sequence specific. So that's a bit the mechanism.
Thanks for that, because I was looking in terms of the high mutational rate that occurs in high mutational rate that there should be an element of conservation or conserved sites that would actually relate to its integration. But as you say, it's not really very site-specific.
Actually, it is a good question because we have actually a paper on this. But now we go really in detail. So for the general sake, we say no real sequence specificity. Still, some amino acids of the integrase do bind the double helix. They recognize the chromatin, they have to bind. And they bind better to some bases than the other, more hydrogen bridges. And so what we did is we mutated these amino acids that bind to the DNA, and then you see that this logo, which is a kind of preference for the bases, changes. And actually, we found one patient which had this mutation together in your cohort in Durban, and found an effect on the progression of the disease. Yeah, but this is like the very, very fine tuning. Yeah, there may be something, but that's kind of like the really, I mean, the spices on the herbs, something like that. But in general, we say no integration specificity. Yeah.
Any more questions? Dr. Answer.
Thank you for a very intriguing. I just want to ask a question on. We know that after HIV infection, and we know that the immune system is chronically activated. And then that is actually the hallmark of a lot of comorbidities, like CVDs and cancer and so forth. So I just wanted to find out, did you perhaps explore if the so-called legends, if they somehow either silence or totally eradicate chronic immune activation?
Okay. It's again a good question. It's a bit a double-edged sword, I think. It's not only chronic immune activation, it's also the chronic inflammation. It's not only specific immune, it's just the non-specific inflammation which is activated, which indeed probably gives premature aging and a lot of complications. And it's definitely worth studying. I mean, in theory, we didn't study that because we cannot study inflammation in a cell culture. I mean, yeah, you could mix cultures and so on, but it would be, I think it's something eventually to be tested also in the patients if we do these things in the patients. Theoretically, we block expression of the proteins, we put them in deep latency. So if the protein expression or it can even the transcription, the mRNAs, it shouldn't always be proteins present on the membrane, it shouldn't always be viruses produced. It could even be that short pieces of mRNA already induce inflammation. If we completely block transcription, we would solve that. Yeah, that's half of the answer. The other half of the answer, and it's something which I've been discussing recently, if you see these natural mechanisms of selection of the actively transcriptional active cells which are killed, so the negative selection, it means that our immune system is still doing something. And so blocking all this activity may interfere with the natural selection mechanism. So, of course, if our block and lock immediately blocks all transcription and we can cure, then we don't bother. But if our interference, whatever, it could be another, interferes with this natural selection, then we may be doing something bad. So it's in that light. But theoretically, a complete block and lock would affect transcription and inflammation. it would be a solution for that as well. Yeah.
Thank you.
Thank you, Dr. Hansen. Is there anybody that has a...
Ask a question.
Okay.
Ooh.
Thank you very much for an interesting talk, Dr. Peter. You probably did mention this in your introduction, maybe I missed it. What makes you think or being optimistic with the functional cure or the block and lock strategy as opposed to the other strategy that you discussed, which is purge and lock, purge and kill,
Purge and kill.
which I think it seeks to clear all the infected cells in the person living with HIV?
Yeah. Well, again, the answer will be composed. I deliberately didn't tell that one strategy is definitely better than the others. I said we should try different things, let 1,000 flowers bloom. It's good that people approach stem cell transplantation, gene therapy, purge and kill, and block and lock, because in the end, what will work will work. I'm a bit critical about the purge and kill because it has already gone through many clinical trials and didn't meet expectations. Block and lock, we didn't do a clinical trial. Okay, you can say you don't even know if it works. That's true, but also we don't have negative clinical trial data. So as a scientist, you have to be optimistic. Optimism is a moral duty for a scientist. I mean, if you're not optimistic, then you better stop your science, your research. I mean, it is a lot of effort and pain to do the research, so you really should go for, you should have a goal, and you should believe in the goal. So you should be ambitious. My main saying about Legends is that I think it should be tested. There should be definitely a clinical trial to test out whether it works or not. If we do not test it, then I would be like dissatisfied. I would be disappointed. But whether it would be working at the clinical and the patient level and the consequences, yeah, that's empirical. That's really like do the clinical trial. And then the last part of the answer is that because we got a question also yesterday, that there should be no animosity between the purge and the kill and the block and the lock people and saying like ours is best. I mean, we are exploring in the lab combined strategies, and that people have proposed, yeah, why not combine? Why not block part of the virus in deep latency, and then the ones that you still, the small peaks you still see, that close to the enhancer, maybe these are the ones that you can activate, that you purge and kill, and maybe you can kill those with an LRA. And then a combination, like a block and shock, or a block and kill, maybe that's the way to go. And that's definitely something we can explore as well. Where you retarget, and where then during a short period you try to reactivate the ones that you could not retarget and kill those out of the body. And then that would be, I mean, that would be a solution to what we see of this residually transcriptionally active proviruses. It would be nice, then everybody would be happy. Yeah.
Okay, we got still 5 minutes left. So,
We can proceed.
okay.
Thanks
Okay,
for the
Mike's
talk.
quarter.
If you have to take the animal models, the introduction of the legends, it will not be recognized as a foreign substance. And then the body itself starts to fight with the legend instead of it solving the HIV virus.
No. It's I mean, this is the legend of a small molecule, yeah, like you have any drug. So I don't think it will induce any immunity against the drug. This is very unlikely. Small molecule. It's like an hapten. It has no carrier. So I mean, what you say would be true for any drug we take. So that's not the case. The question may be why we didn't do the animal model. We've been thinking about it. At some time we were planning. We didn't get a grant. It is not easy because you have to optimize then the molecule to give like in a mouse model. You have to do PK studies, which we do not have because we have PK for humans. In the animal model, it's actually a culture flask with a fur and a tail, because it's very artificial. You take human cells, you infect them, you put them in a mouse, and then you would add your legends, but you would be definitely too late because you already infect your cells before you add your legends. So it's very complicated to do it. And so we actually stepped down from the idea, and we know since people, I mean, that I didn't tell, but there is one company that now is proceeding into a phase 2 with a legend. They want to test it as an antiviral, and I'm trying to convince them to also do a clinical trial for a cure. And so it was tested in a phase 1, it was cleared, no toxicity, no problems. So they're going to go in phase 2, and then they can measure integration sites, they can measure reactivation, so they could test if it also have effect on a cure in the patients. I mean, because the study will be done anyway. Yeah?
Okay. Is there anybody still? We got still 3 minutes left. Yeah. Who? Prof. Mamfuda?
Okay.
Yes.
I wanted the students to actually be the ones who are asking the questions. Just a general question out of interest. Integration is more of a late or mid event in the life cycle of the HIV. In your own opinion, is it the most ideal therapeutic target amongst the other stages of HIV cycle?
Yeah.
What's ideal? I mean, I only can say from practice that the integrase inhibitors, and we're not talking about legends, I talk about dolutegravir, bictegravir, they were the latest to be developed after the reverse transcriptase and the protease inhibitors, and they're now the most potent antivirals we have to date. Is it because it's the best target, or since they were the last developed, they had to, I mean, they were competing with ones with existing, so they had to be made better? It's difficult to say. I mean, for one aspect, definitely, is the resistance. They have a very high threshold for resistance compared to the other targets. But that's also something empirically found. It was not predicted. We now have lenacapavir, which is a capsid inhibitor. It's given in certain circumstances. It's a long-acting drug. It's a bit prematurely to say how it will act compared to the integrase, whether it will be even more potent. It's too early to tell. But from clinical practice, at this moment, the integrase inhibitors are the best. But yeah, they also took the longest time to be developed, so it's a bit double.
I think we're going to take one more question, and then maybe I would like also just to ask an open question after that.
Okay.
Is it fine?
Yeah.
Okay. Dr. Answer.
It's me again. I just want to ask, maybe it's a theoretical question, I'm not too sure. You mentioned about the GPS. I'm not too sure you're referring to the LEG-GF will act as some sort of enhancer to direct the virus to the genome.
Yeah.
If LEG-GF was perhaps manipulated to a different region in the genome, do you still find the same effect, or is it just unique to that specific site where?
Yeah, very good question. Actually, we did that. Years ago, we actually made fusions of LEGF, where we replaced the PWP domain with other DNA binding domains. And so we retargeted, you can retarget integration by doing that. Yeah? Did we get deep latency? No. But the domains we took were, I mean, we didn't retarget, I mean, we did these experiments not to prove a block and lock, but just to prove that we could do retargeting. Yeah? So even with these domains, the virus was active, but retargeting we definitely could do. But it was more a concept of a gene therapeutic approach. Yeah. Yeah.
So there was no effect that you guys found? There was nothing, did it enhance the integration?
No, it didn't have effect on the integration efficiency. It affected the site of integration because, I mean, it was at a different site. Now it becomes technical. We didn't do that with the full-length HIV. We did it actually with a lentiviral vector, and these lentiviral vectors have a heterologous promoter, CMV promoter. So they were active, but they were not driven by the LTR. So we didn't do a retargeting with an LTR kind of virus to see if we also could retarget and see what the effect would be on the transcriptional activity. Yes, theoretically you could do that, but I think with our legends we proved that in a more elegant way, no? Without disturbing less in the cell. Because if you do this retargeting with LegF, you also affect all the other binding partners of LegF. They will also be retargeted. So you not only affect HIV, but the legend, since it's bind integrase, it's only affecting HIV, so it's much more specific.
Okay, maybe I thank you, audience, for your interaction. It was very, enriching. Maybe, yes, I will ask two questions, one for the students but one on science, perhaps.
Okay.
I just want to ask you, what is your take on the elite controllers? Why don't we take advantage, perhaps, to study why the B cells are able to keep up, with the pace of the virus mutations? Do you think that might be an option, or what?
Okay, but we do study elite controllers. Actually, we have a paper together on elite controllers, but one of the problems is that elite controllers is a very heterogeneous population. There could be many reasons why you become an elite controller. And many different aspects in the immune system. I think it got renewed interest. Yes, I think Matthias Lichterfeld, I think he saw the P21 protein. So people have said that some host factors, restriction factors, which may be different from patient to patient. We had the idea that maybe in the African population you will have different, because genetically we're different protection than in the Caucasian population. It's all possible, but the studies as far I know is that they always come up with some other features, and it's probably a mixture of different things. But it's being studied, and I think people, and with this Lichterfeld study, it's a renewed interest, because they can do fast what otherwise takes 20 years.
Right, and then of course we also, I am sure you students, and maybe you have mentioned in the beginning of your talk about the BELSPO program. So I just wonder if you can just maybe give a slight thing in what is the in for postgraduate students, etc.
What is it?
What is the in for them?
Okay, but I think I mentioned it already in the beginning. So the BELSPO is a research network between 3 labs and groups, and one of the aspects we will do 2-3 years is training students. That will be done in the different institutes. So people from Leuven, from my lab, will come to train people in HIV science, HIV cure, specifically the cure, the effect on the subtype C strain, which is prevalent here. So we will do a workshop on that. And then we will also exchange students between the 3 different labs. So people can be sent to our lab, where they trained in integration site sequencing some of these aspects. So it's still the beginning. It's the first obligation, or the first, possibility of this grant that I'm fulfilling now. So I'm needed to come to talk. with the people here to see the different labs and talk also with the students. Also ask a bit about expectations and possibilities. And while we continuing the project, we will train people, and training can be indeed in a workshop, by lecturing, by discussing with students, but also by exchanging students. Who we will send, how long, how we will do it. It's some still in the thinking and in the process, but definitely for people who want to pursue a career in HIV research. I mean, I should say, I mean, at least you should have an interest in scientific, to do scientific research on HIV. If you just say, I want to go 1 month abroad to travel nicely to Leuven and eat our nice chocolates and drink our beer, that's not the right motivation we're looking for. But we have the possibility.
It's part of the program.
It's on top. It's a little bit on top. But the main motivation is for students, and there should be students here in South Africa, because it is definitely still a problem, that are motivated to pursue a career in HIV research. And I know there are different levels. Finish your master, start a PhD. And for those, we can offer this training, exchange, and discussion with PIs. So that's what a bit the BELSPO network wants to do as a start. Okay?
Thank you, Prof. Ziege.
And so students that are here and motivated, I mean, feel free. I don't know what the rest of my program is, but if you're inspired by this talk and you have questions, I mean, we have the World Wide Web, and we are all internet and connected. Just, I mean, you find me on social media, Twitter, email, whatever. You contact me, we can talk, we can discuss. So it should not be only on these fixed occasions, but we are open for the discussions on HIV with all of you. Yeah?
Yeah.
And I wish you all success in your careers, whatever you want to do, and wherever your ambition is.
Thank you.
Okay.
Thank you, yeah. Thank you, Prof. Prof did avail himself maybe after this talk, especially for the students in PEH, Physiology and Environmental Health. If you still want to come and talk with him in the wet lab, you are welcome, okay? So the last thing now, we need to end, and we're gonna call Dr. Hansen maybe just to give a word of thanks, and then we closed.
Good afternoon, everyone. From the Faculty of Science and Agriculture, the School of Molecular and Life Science, and very specifically the Department of Physiology and Environmental Health, we would like to thank our honored guest, who is Prof. Ziege de Buyse, for his time that he has spent flying from abroad till this rural lands of South Africa, just to give us a special talk on a very important topic, finding a cure for HIV. Lastly, I also would like to thank you as the audience who have made it your time, or made your time to be here with us. And specifically, a special thanks to the acting director, Prof. Mampuru, who was here with us, and a special thanks to our HOD, Professor Van Staden, for her time, and also everything that she has put in to make this event a successful event. We really thank you all. Thanks.
Right. And now public lecture is officially closed. Thank you.